All right, so these are our notes for section 3 of chapter 3 in biology. We're going to talk about the cell membrane in this section, the structure of the cell membrane, and then we're also going to talk about how things cross the cell membrane and how you can send chemical signals across the cell membrane. So, a couple definitions to start out here. Just like always, fill in your blanks. A cell membrane is a thin phospholipid bilayer that separates the inside of a cell from the outside. So again, definition itself says phospholipid. You kind of need to know what that is, right? That phospholipid bilayer, right? That's what we're going to do on the next couple slides. You have these phospholipids specifically, and we talked about these in the lipid section of last chapter, right? So we have this polar head group, right? That's this phosphate and glycerol, right? And then we have these two non-polar fatty acid tails that are connected to that head group. So I got 2 fatty acid tails. Right? Again, this half over here, right, is very polar, right? That's my polar part. Whereas the non-polar part of this is are my tails, right? So be able to draw a picture of a phospholipid, understand which part is polar, which part is non-polar, right? And this polarity is actually what creates the structure of our cell membrane to begin with. So it's really important that you understand that there's 2 different parts here. This is actually called an amphipathic molecule because it has polar and non-polar parts. So that's important in terms of the structure. So we jump to this next slide here and we start talking about these micelles and bilayers, right? So since a fatty acid, again, polar head group, non-polar tails, right? Since it's polar and non-polar, it tends to take on certain structures automatically when you throw it in water. So if I threw a bunch of these molecules together in water, what they tend to do is create a micelle. And a micelle is actually just a spherical structure. So I drew a circle here, but it's actually a sphere. I understand it goes all the way around in all directions. And you got these polar head groups on the outside, and you have these non-polar tails on the inside of this structure. So if this ball was a basketball, the surface of the basketball is covered with these polar head groups, right? So again, polar, non-polar. I have these polar head groups on the outside of my sphere. Sorry, I'm using a little tablet to draw on. It's not always easy to do. And that polar head group is actually attracted to the water that's surrounding this micelle. Whereas the non-polar tails that are in the center here, they are actually repelled by that water. They're attracted to each other, and they're what we call hydrophobic. They're actually, that means that they're scared of water, essentially. So they stay in the middle while the polar head groups stay on the outside. And this is automatic because polar goes with polar and non-polar goes with other non-polar. Polar and non-polar don't mix very well. So this is an automatic structure that forms when you throw phospholipids into water. A bilayer, though, is just the next step up, right? If I made that outside of my sphere big enough, right? I'm gonna have a lot of phospholipids in here, but you kind of get the gist of it. I'm spacing them out a little bit too far, but I don't want to draw a million of these things. But if I made the outside layer big enough, and you got these little tails hanging off to the inside, right? Everyone has 2 tails, but I get kind of lazy eventually, right? What can happen is, again, I have this water on the outside that the polar head groups are attracted to, but I'm also going to have some water inside of this structure now, right? So I don't just get one layer of phospholipids, I actually get a bilayer. I get 2 layers, right? But on this internal layer, all of the polar head groups are pointed towards this water on the inside. Again, polar goes with polar, nonpolar goes with other nonpolar. So these polar head groups are attracted to the water that fills this bilayer structure. There's water on the outside that my other heads in my bilayer are attracted to, right? But on the inside of this structure, I have tons of these nonpolar fatty acid tails, right? So again, there's two per thing, but it's kind of hard to draw those eventually, but they go all the way through here, right? All those fatty acid nonpolar tails are the inside of that structure. Again, kind of a sloppy drawing, but you get the idea, hopefully, right? Nonpolar stays on the inside of that bilayer, and that bilayer is what is essentially a membrane for us, right? So don't worry about if your pictures are sloppy here. We'll draw a good membrane here in a second, hopefully. I'll try to. But that bilayer structure is automatic. As soon as you throw phospholipids into water, they do that. So that membrane that forms around your cells is just a natural occurring structure. I mean, it doesn't take energy for your cell to put membranes together because they want to be that way, right? The hydrophobic tails want to stay away from water, and so they take on these structures automatically. So again, a fluid mosaic model, right? Now in your notes, you don't just have this fluid mosaic model. So if I skip ahead at selective permeability, below this, you should have a list of things. You should have phospholipids, cholesterol, carbohydrates. There's one more thing. Phospholipids, cholesterol, carbohydrates, proteins, right? So you have those things down below. I'm not really going to put those in these notes. You have to pay attention and fill in there because I don't want a definition for those things down below, understand? I want you to take what I'm talking about here as I'm drawing this model. Each one of those things has a job within this membrane, right? It does certain things. And I'm going to talk about those things, but I'm not necessarily going to write all those things down. So you have to pay attention. Excuse me, my dogs are barking at something. Hopefully they'll calm down. I'll just take a timeout here. I can pause. Alright, so sorry about that interruption. So I'm going to draw this fluid mosaic model. The first piece, right, just like we were talking about, the first pieces are going to be these phospholipids. So I'm going to draw a bunch of phospholipids. And there's two layers, right? It's a bilayer. So you got one layer on one side, one row of those phospholipids on one side, one row on the other side, right? These phospholipids are basically going to glue everything together. Because again, we talked about how there's polar and nonpolar, right? There's polar on the surface, right? That's separating two water solutions, one side and the other, because this is a membrane. This is just a portion of the membrane. And understand this extends, right, around in a sphere, around a cell. But those polar head groups and nonpolar tails are taking on that bilayer structure that we talked about before. But as I continue to draw this, I need to add in new things. This is called a mosaic model because it has lots of different pieces that make up the whole, right? So just like if you were to make a mosaic in your art class, right, you take different colored pieces of glass and throw them together to make this picture, right? Whatever it is that you're doing, right? That's the idea here. We have different parts. So we see the phospholipids right now. I need to start adding the other things that end up as part of this membrane. So I got this phospholipid bilayer started here. The first thing I'm going to throw in, and I'm not doing this in any particular order, is a cholesterol molecule. So if I draw in a cholesterol molecule that is a steroid, right? So it has these ring structures that we talked about before. Again, if yours isn't perfect looking, because mine isn't either, don't worry about it. Because I just kind of screwed up that hexagon. Anyway, You get this idea. You have these four rings that are connected together. Mine are kind of separated here, but it's not a big deal, right? On cholesterol, there's this tiny little hydroxyl group. I didn't make that big enough to be visible. Let me try that one more time. There's an oxygen and a hydrogen. That's kind of like a water molecule, right? So I have this oxygen connected to a hydrogen, and you know that that's going to create a polar separation. You know that oxygen pulls on electrons harder than hydrogen. That's why we had the hydrogen bonding last chapter, right? So that's a very polar part of this cholesterol, whereas the rest of the cholesterol is nonpolar, right? It's all carbons and hydrogens. So cholesterol is going to be found in my membrane, just in between phospholipids, right? And understand that the size and scale of this is not necessarily accurate. Cholesterol is actually a fairly small molecule, but it has a purpose. It has a job within this structure. So I'm just drawing some phospholipids to kind of finish off this area, this part of my membrane. Again, this nonpolar portion is in between those phospholipid tails, those fatty acid tails. And what it actually does is it makes this membrane fluid, right? When I say fluid mosaic model, I mean that you can have all of these molecules within here can move around each other. They're not actually static. They can rotate, they can move, as long as nonpolar stays with nonpolar and polar stays with polar. So I keep going with this structure, right? All the polar things are on the surface. All the nonpolar things are in the center of this membrane, right? As long as that's true, all of these molecules are free to move throughout this structure. It's a fluid mosaic model because molecules can actually move, right, freely throughout this structure. But rules have to be followed. Nonpolar has to stay with nonpolar, polar has to stay with polar. So what cholesterol does, and this is what you put below, remember, is it makes that membrane more fluid. It allows these molecules to continue to move freely from one another, because if you remember from notes last chapter, I had another lipid that looked like this, right? And that's a fat. Whereas if it was an oil, it would have kinks in those tails. And when you have a fat, the difference between a fat and oil when you're cooking, is that a fat is going to be a solid at room temperature, whereas a oil is going to be a liquid. They're both lipids, they both have similar properties, but one's a solid, one's a liquid for a reason, right? If you have these tails here, and the only difference between a fat and a phospholipid is that it's two tails instead of three tails, right? They're still straight tails. If you have structures like that, they actually can stack really close together and they can take on a more solid state, right? Now, the oil, on the other hand, has tails that are actually kind of bent and shooting out in different directions. And that actually prevents molecules for oils from getting close together. It keeps them from becoming a solid. So again, if I take that same idea here, I have all these phospholipids that are making up this membrane, right, giving me that structure to begin with, polar, nonpolar, right? If they're allowed to get really, really close together, this is not going to be a very fluid structure. It would actually be more solid. Things wouldn't be able to move freely. So cholesterol is in your membranes, in all of your membranes, and it makes it more flexible. It makes it more fluid. It actually makes it stronger in the long run because of that. You can actually bend and move this membrane without tearing it, right, as a result. Because if it was a rigid structure, any movement could actually break it, right? Could tear it apart. So cholesterol makes the membrane more fluid, but it also adds to the strength of that membrane, right? It allows it to be a little bit more flexible. So make sure you put that below. Next thing I might put in my membrane, right? I can draw in a protein, right? Now there are different kinds of proteins. This protein that I just drew in right? And just made a big blob structure. We talked about proteins and how they're made of amino acids, and this is actually just a random shape I drew, but this protein has some sort of function, but this is a different kind of protein than the next one that I'm going to draw. Right? This protein, since it doesn't go all the way through the membrane, since it doesn't span the membrane, is likely going to act as an anchor, right? I'm going to attach something to the surface of this protein, and it could be a lot of different things. It could be the cytoskeleton, so that I can pull on that protein and move the membrane to actually change the shape of my cell, so I could have a muscle cell that actually moves and helps me move my body, right? So it could be involved in something like that, right? It could be attached to the cytoskeleton to allow this cell to move, because you could pull on it and you could change the shape of the cell. But it also might just have some kind of marker attached to it. That's what I'm going to add to it here in a little bit. But again, we talked about that polar nonpolar before. If I have this protein that's floating in this membrane, this part of the protein down here has to be nonpolar to match up with all these nonpolar tails of my phospholipids. So it's going to be in a specific orientation in a specific place. Again, I still have a fluid model. These molecules can kind of move around to different places, past each other, through each other, but polar always stays with polar, nonpolar always stays with nonpolar. So again, sometimes proteins act as anchors. That's what this protein is going to act like, right? Put that down below. Otherwise, other proteins have other jobs. So I'm going to add a couple more phospholipids so I can kind of extend out here a little bit. Again, try to make a decent picture of this. It doesn't have to be perfect, but just have a general idea of what's going on. So I'm drawing a protein here that's actually going to span the membrane, go all the way across. Now this time, understand this is actually a solid protein. I drew two pieces, but I had them open in the middle for a reason. This protein, although just like the other one, having polar and nonpolar match up appropriately, the ends here would be polar to match up with the polar head groups and the water out here. But the inside, right, would have to be nonpolar to match up with these tails, right? But what might not be clear with this drawing is this is actually a channel protein. So it's essentially like a straw, right? It's got an opening going right through the middle of it. And I can only draw a cross section of this, obviously, because I'm drawing a 2D picture. So really this is actually like just a portion of this protein that you're seeing. But it has this channel right down the middle. And so on the inside of this channel, there actually would probably be polar. That protein would likely be polar, because what's going to happen here is I'm going to have something like maybe a water molecule that's very polar, right? That can be transported through this channel protein, right? It can go from one side of the membrane to the other, but without that channel to go through, because that water is very polar, that water wouldn't be able to easily go through the middle of the membrane, right? It can't go through this nonpolar portion of the membrane over here. So I drew a channel protein here. It helps transport things across the membrane because it spans the membrane and it's got an opening in the middle of it, right? So again, down below you got proteins actually have two different jobs. They can help transport things if they're channel proteins. They also can act as anchors, right? And then the final thing I need to put in here, because it also talks about carbohydrates down below, right? If I have carbohydrates, right, attached to an anchor protein like this one, right, they can also be attached to different kinds of, they're actually called sphingolipids, so that's actually kind of a confusing thing. It's above level for us, don't worry about it, but they can be attached to proteins as well. So I'm going to attach a glucose, a sugar, a carbohydrate to the surface of this protein, and I'm going to have like a chain, some kind of structure here that actually acts as a marker that can help me identify this cell, right? So you have blood cells in your body that are a specific type. You guys have heard of A, B, O blood types. Well, the reason why your body knows whether that blood cell is yours or not, right? The reason it would know to reject some cells and accept other cells as your own and not attack those things as a result, right? This is an immune system issue, right? You have markers on the surface of your cells, right? These glucose, these carbohydrates that are on the surface of the cell here. And glucose is polar, like water, so it's okay that it's up here, right? But the purpose of it, the reason that carbohydrate is there, is it helps you identify that cell. It helps your immune system determine, is that my cell or is that a different cell? Because if it's a foreign body, if it's a cell that came from outside of your body, you might need to attack it because it might be dangerous to you. Maybe it's a bacterial cell that's actually very, very dangerous, right? Could it make you really sick? So markers on the surface of your cells like this are made from carbohydrates, and they help you identify that cell. So that's the job for carbohydrates in a cell membrane. So again, you're supposed to have jobs for all parts, right? I can go through that real quick. Phospholipids, right? They provide that basic structure, the polar, nonpolar, right? They hold everything together. They're the glue, right? Cholesterol made things fluid, right? It actually made the membrane a little bit more fluid. It made it more flexible. It actually strengthened it that way. Proteins, if it's like this one here, they can act as anchors. Sometimes they're transport proteins, they're channel proteins that span the membrane and help things move across that wouldn't otherwise be able to move. And then carbohydrates help you identify a cell. They act as they're markers on the surface of the cell that you could use to identify that cell.